All right. My name is Tom Keller, and I'm a co-investigator for Build Exido, and I want to officially welcome all of you to the 2021 Build Exido Symposium, where we have the featured presentations for our students who have just finished their summer immersion or their intensive summer research program. And going to be sharing with us the research that they've conducted. I'll make note that we have a new training program called URISE at PSU that is also an NIH-funded project. And we have a couple of students who are in that program are joining us today. They started in Exido and then went into URISE, and they sort of accelerated and went into the immersion in their very first month of the program. So they have not been in RLC labs for a full year and are going to be sharing research nevertheless, probably their initial proposals for a project coming up. So we want to encourage them to participate, even though they're not as far along in the process. So this is an exciting time for us to learn about all of the great work that you've been doing. And we also realize it's a chance for you to gain experience as a presenter, sharing your research. And we know that that can be anxiety provoking. Maybe you're a little nervous. That's natural. Everybody feels that way. But I'll just remind you, you got through your elevator pitch, which probably gave you the same feeling. And that's something you can, you all succeeded in doing, and I'm sure you'll get through this just fine. So we want to encourage you to, give yourself a break to yourself and your peers. You might make a mistake, but just take a breath and move on. We want you to, of course, be respectful for each other. And as my child's kindergarten teacher said, we want to have, we welcome put-ups, but no put-downs. So just a little bit about procedures for how we conduct this. We have a schedule of presenters. We have about 10 minutes slotted for each one, which includes a little bit of time for question and answer afterwards. So I'll call the name of the presenter, and you can go on video if you're comfortable doing that. And you can also share your screen to present your slides. And as you're getting started, if you can just say hello or hi, so that the Zoom will know that you're the speaker, and the yellow box will be highlighted for you, and then you'll be featured along with your presentation in the recording. So we are recording these so that you'll have a chance to see the presentations given by your other cohort members. Those will be posted in about a week. And you can direct your family and friends to see them as well. So as you're getting started, again, say hello. And when the box lights up for you, then I'll do a thumbs up, and that means you're ready to start. And then you can give your presentation, and then at the end, I'll invite any questions people may have, and we'll go through that routine and try to stick to the schedule as well as we can, recognizing that we're getting a little bit of a late start. Does that sound good? Any questions? All right, so our first presenter is Dimitri, who's been working with Dr. Goforth.
Yes, hi. I will try to share a screen. Somebody let me know if that's working, please.
That looks good. You're ready to go.
Awesome. All right. Hi, my name is Dimitri. I've been working in the Go Forth lab towards a project we're calling silicification of lipid nanocarriers. So to start with, I would like to talk about a little bit about why, there we go, why we care about lipid nanocarriers. And so here are just some examples of the things they're used in. A lot of these are like targeted drug treatments, such as like cancer treatment, chemotherapy, where we want things released in a specific area of the body. And others are things like the new Pfizer and Moderna coronavirus vaccines, which are also based on a different type of lipid nanocarrier. And so to sort of build on that, we can talk about the different types of lipid nanocarriers that there are. On a very basic level, there are these micelles, which are sort of these one-layer lipids with polar head groups and non-polar tail groups that form these little balls that mostly have just space for non-polar molecules to fill the inside of. Then there are liposomes in the middle, which have a little bit of space for non-polar particles to fill, but mostly are usually filled with polar drug particles of some sort that can be delivered in the body. And then there are true lipid nanoparticles, which are more like what the new vaccines use, where the polar groups are sort of stored in their own individual bubbles of lipids inside of a larger molecule, or not molecule, a larger structure. And so these have some issues with stability. And the issues that they can have at higher temperatures, or really room temperature, is one issue is aggregation, where you can take these individual lipid nanocarriers and they can start to clump together and form these large clumps that fall out of solution. And the other issue that they can have is they can start to fall apart and decompose. And either way, the expected or sought out outcome does not happen, because they're not able to deliver their payload the way that they were intended to. So first, in order to work on the silicification of lipid nanocarriers, we had to synthesize some lipid nanocarriers. And so what we had to do was synthesize some liposomes. And we chose liposomes partially because they're sort of the middle of the road between the three types of lipid nanocarriers that we have. And so this showed a little bit of our procedure of how we did that, forming a lipid film on the inside of a round bottom flask, hydrating it, and then sonicating it to get it to a more regular structure. And then this was our DLS data that showed that we had a sort of normal distribution of sizes in our liposomes. This picture here was the film. It wasn't very regular, but it did show up, and it did end up after sonification giving us a fairly normal distribution of liposomes. And so then we can talk about silicification. So why did we choose to use silicification on these liposomes? And that is because we saw through Dr. Stedman's research and a little bit talking to his lab that he was able to coat viral capsids in silica and then bring them to temperatures where they would normally be unstable. And what happened was that they didn't end up destabilizing or denaturing. And when he brought them back down to conditions where they were survivable and removed the silica, they were still biologically active. And so using that information, we decided, we weren't supposed to, there we go. Using that information, we decided to try our own silicification technique based on one somebody in our lab came up with, where they added a silica coating to silica nanoparticles. And we haven't gotten to this step in practice yet, but I have done this myself, this procedure with these silica seeds, and working on the coating so that I know how to do it, and we will soon be applying that to our liposomes. And so that'll look something like this, where we have that liposome solution that I showed earlier, and we will be adding the silica to it and hopefully generating some silica coated liposomes for us to work with from there. And so the idea behind this is that hopefully they will show the same increase in stability that Stedman's viral capsids showed when coated with this silica. And so we sort of have developed our silicification procedure for our liposomes. But once we do that, we will be able to determine the effectiveness of the silicification at stabilizing our particles through DLS and weight and density data, and continually run those until we have some idea of how well that has worked. Yeah, and that's my time.
Great, thank you. It's very interesting, and it seems like it'll have some really important practical implications. Any questions for Dimitri about the presentation? So I'll ask one.
All right.
So you mentioned that the impetus for this project was research done by Dr. Stedman in his lab. Have you been collaborating with them on this procedure, developing those procedures?
Actually, we have a little bit, and he's here as well. I saw his little tag pop up. But yes, he has helped us quite a bit with deciding how we're going to go about this and sharing his experiences.
So is this an example of basic research being converted into sort of a next step towards an application?
Yeah, I would say so. I would say it's still pretty basic, and that, like, you know, we're just kind of trying to see if we can develop these materials and what will happen. But there are more easily seen applications to this step.
Yeah, a good goal would be to increase shelf life of the vaccines, the coronavirus vaccines.
Yes, that would, yeah.
Keep
be a...
Yeah, it's disheartening to hear about so many doses getting, you know, discarded.
Mm-hmm.
Yeah, my mom works in the medical field, and she tells me all of these stories about how, like, something accidentally got left on the counter for 15 minutes, and now they have to throw it away because they don't know exactly how long it was there.
So can you give a sense of how far along you are in the research? I mean, in the project overall. So you've developed the procedures. So going forward, when do you expect to see if this really results in the kind of extension of
Yeah, well, going forward, I am hoping / expecting to try our first silica coating procedure within this week or maybe next week. So we'll sort of get some preliminary data on that very shortly.
Okay, so then it's just a matter of, like, what's the time scale you're trying to look at for how stable they are?
That's going to depend on a little more analysis of our initial particles and seeing how stable they are and how quickly they fall apart. I know with the coronavirus vaccines, they tend to only maintain their stability for about 2 hours outside of a refrigerator. So in that time span, they would normally degrade. But since we're not using those exact particles, we'll have to base it on how long our particles do last and how much we can extend that shelf life through silicification.
Right. So this could be just a matter of weeks or months before you see if this is yielding results.
Yeah, hopefully.
Any other questions for Dimitri? All right. Well, again, we appreciate a wonderful presentation. Thank you.
Good job.
So now we will move on to our next presentation. Next, we have Kim Paulin Jr., who's been working with Dr. Sanchez.
Yeah, hello everybody. So I've been working with Dr. Sanchez, but I am going to be sharing my research that I actually did with Dr. Marilyn Makowitz before she left PSU. This summer I've been working a lot with Dr. Sanchez, essentially getting started on a new project that he's going to have for me, but it's been kind of hard to do it since we're not in the lab. So I've been working a lot with Dr. Makowitz in actually writing a research paper. So I'm just going to be presenting the data that I was able to collect beforehand and the paper that I've been working on. So you might have seen this presentation before, Dr. Goforth, because I think you might have been at the pieces from last year, but it's the same paper and I've just been working on the paper itself. So I'll go ahead and just get started. So this is going to be on the design and synthesis of oxidation-resistant nickel nanoparticles. So we will begin with what are nickel nanoparticles. So nanoparticles themselves are a very small material. They're on the range of about 1 to 100 nanometers in size, and they each have, all nanoparticles have specific properties in comparison to the bulk, including a high surface area to volume ratio and different electronic properties that are very different than the bulk, including characterization by UV spectroscopy. Nickel itself has its own properties. It's a group 8 transition metal, which makes it highly conductive and magnetic. But the important part is that it's highly redox active, which means that it readily exists as a nickel 2+ ion, which is highly toxic. Some applications of nickel nanoparticles include catalysis and electroanalysis, but we're focusing on biomedical uses. So we actually are creating this particle for a biomedical application that we won't focus on in this presentation. We mainly focus on the synthesis and design of it, but there are several different uses already out there. So here, a different group actually did a toxicity assessment of the nickel 2+ ion leaching. And when you look at this graph here, you see that the comparison of that nickel 2 ion has a much lower required concentration for that LC50, or that lethal concentration of 50% of the population, in comparison to the nanoparticles themselves, which basically tells you that it is a highly toxic ion. Other groups have actually been able to synthesize nickel nanoparticles themselves to shield that oxidation effect of the nanoparticle. And here you see they dissolved nickel in comparison to the nickel nanoparticle in solution itself. So you're able to look at how effective this particle shielding is, which it is fairly effective. However, there are some major drawbacks which are common with nickel's nanoparticles themselves. It goes more into detail on it, but I'm going to go ahead and skip over this for time's sake. But some synthesis methods that we see in other applications or other methods that are out there include the solvent system is typically organic, which is not very green. Its reducing agents are typically fairly dangerous. Hydrazine is a notable one that's actually a component of rocket fuel, and it requires significant heating, typically between 60 and 250 degrees Celsius, if not higher, which is rather energetically consuming itself. So major challenges of nickel nanoparticle synthesis include solubility in water, synthesis at room temperature using non-harsh reducing agents, and the preventing oxidation of nickel itself. So our major question we ask is how to synthesize oxidation-resistant nickel nanoparticles at room temperature in an aqueous environment. So in our lab, or in Dr. Makowitz's lab, we actually previously used this similar design. We used metal colloids, iron, silver, and gold were some that we've used previously. And then we're surrounded with a lipid bilayer to create that shielding effect that Dimitri talked about earlier. We are then able to tune the surface chemistry with different ligands to customize it to how you would like the nanoparticle to operate. And files are able to anchor that lipid bilayer and those tunable ligands to the metal colloid itself. Here we use a few different molecules itself to actually create the nanoparticle. We use an aqueous solvent and room temperature heating, so it doesn't require much heating, but we do still use this stir plate here. Tunable surface chemistry, we're using DOTAP and sodium oleate as the surfactants to induce solubility in water. We then have a lipid bilayer. Phosphatidylcholine is going to be the main lipid used. The metal colloid is from nickel chloride, and the thiols use are decanthiol and mercaptone decanoic acid, or MUA for short. We synthesized 9 different coatings, and we're going to be comparing the effectivity of all of them. They vary in complexity from a very simple single molecule to a far more complex SOAPC DOTAP DTU with 4 different molecules surrounding the colloidal nickel itself. We then move into characterization. So we find that nickel nanoparticles have this character UV-vis spectrum with a primary, secondary, and shoulder band shown here. And then we have TEM analysis of them, which they typically find, we found that they're typically about 5 nanometers in diameter across. but they vary somewhere between 2 and 10 on average as well. After stability, we look at stability in the presence of oxidizing agents. So we use that well-known method of potassium cyanide in solution, which will induce that nickel 2+ ion if it becomes exposed to that metal nickel itself. And so unstable particles will show this kind of UV spectrum here, where it essentially degrades into a flat line, whereas a more stable particle will remain the same after the presence of KCN. So we're looking at that shift in optical density is what it comes down to. So we find that all of these particles here on the left are highly unstable after presence of that oxidizing agent, whereas the particles on the right here are highly stable in presence of oxidizing agent. We then further assess stability in saline environments. So looking at concentrations of salt, since it is going to be used for biomedical purposes, the salt and pH are both highly important in these studies. So we're looking at shifts in optical density in various concentrations of salt. And we find that the SOAPC DOTAP DT particle and SOAPC DT particles remain most stable across all the salt concentrations. Similarly, in pH environments, we look at pH 2, 5, and 8, since uptake methods can become fairly acidic when taken up into the cell. SOAPC DOTAP DT and SOAPC MUA are actually the most stable below pH 8 when looking at pH studies. We then look at long-term stability studies. So we look at first UV vis. So over time, those particles will begin to degrade. And so we're looking at 24 hours, 1 week, and 1 month immediately after synthesis. So after 24 hours, 1 week, and 1 month, we find that the SOAPC DOTAP DT particle and SOAPC MUA particles are most stable. We then confirm this by ICP-MS analysis. This is actually done at OHSU, so we kind of just hand them off the sample and they look at it themselves. And so same thing, 24 hours, 1 week, and 1 month. We find that the SOAPC DOTAP DT particle, the SOA DT and SOAPC DT particle have the lowest release of all 5 nickel nanoparticles tested, and the SOAPC DOTAP DT will soon be tested, but we suspect that this particle will also remain stable by ICP-MS analysis. So in conclusion, we find that immediately after synthesis, these particles are eliminated because they are unstable immediately after synthesis. And then after addition to KCN, they have we look for minimal shift in optical density, which these particles then become eliminated with a heavy shift in that optical density. Then we're looking at the saline and pH environments, which eliminate this particle here, since it was not stable across various saline and pH environments, and then degradation over time. So PCDT particle actually becomes highly unstable over time, which would not be ideal inside the body since it will release that nickel 2+ ion. So the SOAPC DOTAP DT particle is the most stable over time. So this is the particle that we'll be using moving forward. So in the future, we are going to further tune that surface chemistry of the nanoparticle itself to target subcellular organelles and in order to locate metal pools within the cell, which is the target of this application itself. We'll also be conducting XRD analysis to confirm that the colloidal nickel itself is indeed nickel 0 and not that nickel 2+ ion. So I'd like to thank Dr. Marilyn Makowitz. She's my mentor, principal investigator on this project, as well as Eric Engstrom, who's worked with me very closely on this project, and Daniel Schwartz, and other research assistants in my lab whose undying support I could not have gotten through it with. I'd also like to thank Dr. Sanchez for being a point of contact at PSU for me, and I'm able to talk to you in reference to writing the paper and anything I might need. As well as Portland State University for providing lab space, built Exito for basically training on me how to do research through these years, and the ANAH for actually funding this grant alongside Dr. Martina Riley at OHSU and Craig Barnes at University of Tennessee. So I thank you for your time. If you have any questions, please let me know.
Great, thank you. Jam-packed presentation.
Yeah, it's a big one. I tried to speed through it.
Any questions? So I really appreciated how you summed up with. I mean, it was really a process of elimination. You started with several options and then put them through their paces and found one that kind of performed in all of those situations. So
yeah, it was a very long process. It was back when we were able to actually be in the lab. So I was very fortunate and grateful for being able to do that. But it took like almost an entire year to actually be able to eliminate all of those different particles and do the stability testings and repeat the results. It was a process, but I'm very glad that we got there.
And I guess you're really relieved that at least one survived. What if all of them had failed?
Then we'd have to go back to the drawing board, kind of assess that fundamental chemistry, look at what's going wrong, why are things failing. We had a problem where we are actually seeing aggregates of the lipids themselves with one of our particles, and we were trying to figure out why it was happening because it was essentially the same thing. Just instead of DT, it was MUA, and we're trying to figure out what the problem was. And I believe what we came up with was that the MUA has a lot smaller chain, or that like little lipid tail at the end of it. So that was not making the particles as stable as DT was in solution.
So, and you pointed out the parallels between your work and what Dimitri presented. So this seems to be a particular area of interest at PSU with a lot of research. Is that fair to say compared to, I mean, how does?
I think so. I know in the Department of Chemistry, there were 3 different members who were focused on it. Dr. Mack before she had left, Dr. Goforth, and Dr. Renovare. I think those are the 3 faculty that work in the realm of nanomaterials. Dr. Goforth and Mackowitz like both specifically work towards this kind of aspect of things. And so it's pretty prevalent, I would say. And it's definitely an up and coming field. It's becoming a lot more like major role. And like Dimitri was saying with the whole COVID stuff, like it's becoming like a really critical piece in medicine. So it's really interesting, cool to be like on that cutting edge of science.
Thank you. Any other questions for Kim? All right, another wonderful presentation.
Thank you.
Thank you. So now we'll move on to the next presentation. So Ni is going to present and working with Dr. Peterson.
Sorry, I'm gonna move this. Okay, hello everyone. My name is Dinh, and today I will be sharing the research study I have been working on along with my ROC mentor, Dr. Jamie Peterson. The title of the study is The School Readiness Journey, Capturing the Unheard Voices of Latino Parents. So first off, what is school readiness? So it means children are ready for school and families are prepared to support their children's learning. So school readiness involves 5 developmental domains that work together to support a child's full learning potential. Just to give you guys a brief description of each domain, cognitive development involves how children think, explore, and figure things out. For physical development, the readiness means that they have the gross and fine motor skills to hold a pencil or to hold a book. Language refers to a child's ability to read, write, and communicate with others. Approaches to learning can be described as a child's curiosity to learn new things and engagement in learning. Social emotional is when they are able to start a conversation and interact with their peers and adults and create positive relationships with them. So our targeted population for the study is low-income Latino families because previous research shows that Latino parents do value their children's education, but many feel unprepared and do not know how to prepare their children for school, which can affect their children entering kindergarten, making them more behind than their more affluent peers. This differences in a child's school readiness is also known as the SR gap. In addition, Latino kids are more likely to have skills and learning difficulties as early as 9 months and school age than white kids in the United States. So this is why we need feedback from the community to understand their needs regarding school readiness and develop recommendations for future implementation. For this study, our main aims are to explore the lived school readiness experiences of Latino parents with kindergarten age kids, determine perceptions, usability, and acceptability of primary care-based school readiness promotion interventions, apply design thinking to explore which school readiness approaches or combination of interventions are most desirable to Latino parents within the primary care clinic, and then present key findings to participants, families and providers, and community stakeholders. I have listed some of the school readiness interventions on the side here for your reference, but I plan to move on and will not go over them. So in this study, we will use qualitative approaches, including focus groups, small group survey, journey mapping, and design thinking. Focus groups are split into 2 parts. Part A is about capturing parents' journeys. Part B is about design thinking and mapping interventions along an ideal journey map using the parent stories as a framework. So for focus group A, parents are prompted to explain their own stories with education and then go through their child's journeys up until starting kindergarten. They will end with recommendations for first-time parents and pediatricians. Their stories will be mapped by color-coded. For focus group B, parent journeys are used as a framework to guide ideal map design thinking. Interventions will be presented to parents and get their feedback on several interventions to help parents practice early math reading skills with their children. We will do this at 3 to 4 clinics and then bring the results back to the stakeholders. So as of right now, we are still analyzing the data, but we are expecting some differences between languages, English versus Spanish, and childbirth order at the very least. So some interventions might be more preferable than others, but the perfect fit will be a personalized plan that will include interventions or a combination of interventions that will best fit with the family's lifestyle and needs. In conclusion, the study will allow us to help first-time parents in the future, which will then benefit the whole family and all the children to prepare for school with the implemented school readiness interventions within the primary care clinic. And here are my references. And last but not least, I want to give special thanks to Dr. Jamie Peterson, she's right here, and my team members for guiding and helping me through this research process. And a shout out to BioExido and URISE programs for making this possible. Thank you.
Thank you. Nice presentation. Any questions?
Yeah, I have one. So what was the recruitment like in terms to get the focus group participants?
Sorry, I was mute. We will call, so we basically pair up with a clinic. Right now we are doing it in Woodburn, which there's a lot of Mexican and American over there. So we would tell the clinic to give the patients the consent form and the information about the program or the recruitment. And then if they are allow us to do it, we will call them. And then we will tell them about the eligibility and if they still want to join or if they still want to attend the focus group for the study.
Great, thanks.
So any other questions?
I got a question.
Yeah.
So, I mean, have these families had any kind of support to help the kids to get prepared at all? I mean, through the school district or through the school per se?
Oh yes, as I was there when my mentor interviewed the parents or the patients, and they told me that they had support from the school district to connect their children to Head Start school. So basically they have Head Start as a program to start early and getting them ready for kindergarten. So I think that's the one that the school district has available for the parents right now.
Thank you.
So I'm curious, what field is Dr. Peterson in? Is this like education or pediatrics or?
She's she's a pediatrician, but she's interested in learning about the school readiness disparities and low-income population. Yeah.
So there's a focus on interventions that might be provided by pediatricians, or that the pediatricians would provide referrals if they saw that it would support a family.
Yes, we have the pediatricians can refer the patients to like Head Start or like I said, interventions. I have the library card referral. The pediatrician can refer them to the Head Start program or to the library, or help them figure out how to fill out the application. And I also mentioned Reach Out and Read in my intervention list. That one is to prepare the parents to read and encourage them to read early for their children. Like when they're still like a baby, we will encourage them to read to them and how to talk to them or how to communicate early with them. So that's what that program is doing for the parents.
Other questions?
I had one other one. So you mentioned that there was like some expected differences between birth orders. Can you go into that a little bit maybe?
Yes. So for example, if the parents are getting or are experiencing like if they are first-time parents, for example, they are more likely to be interested in the Head Start school enrollment, which is one of the intervention. And if they already have older kids, they are probably more used to that program or they have known about it. So they won't need that as much. So they might need text messages like to like daily reminder to how to practice English or reading skills with their children.
Thank you.
Yeah.
Okay, any other questions? All right, I'll just note that there's sort of related research happening with colleagues in the School of Social Work in the Center for the Improvement of Child and Family Services, Beth Green and her group. So I'll try to make a connection there because you might be interested in what they're doing on school readiness too.
Oh yes, my mentor is Dr. Zuckerman is my mentor's mentor. So basically I'm working under Dr. Zuckerman, but I'm working with a different doctor on a different field of study, but they are basically connecting, like they are doing the same thing basically.
Great. All right, again, thank you for a wonderful presentation. So we'll move on to the next session. So next up, we have Angel, who's been working with Dr. Thompson.
Okay, so my name is Angel Bowie. I am currently working with Dr. Ann Thompson in her Blue Water Lab at Portland State University. And today I will be presenting the project I've been working on that analyzes the niche partitioning patterns of Synechococcus elongatus. So marine microbes play a crucial role in global biogeochemical cycles and are a major contributor to the marine primary production. Synechococcus will be the focus of this project, and they belong to the Picocyanos genus. They're not as well characterized as their counterpart Prochlorococcus, and they are abundant photosynthetic organisms that show extensive diversity classified in 20 subclades that allow them to be so resilient and adaptive. Their photosynthetic pigment diversity considerably extends their niches that they can colonize. And here's just a picture of the array of colors that they're able to be. A picture of their cell and a abundance global map here of both Prochlorococcus and Synechococcus. The data set used in this project is from the SMILE cruise in March of 2017, and the purpose is to investigate the Synechococcus diversity over depth. This cruise collected 282 samples from 24 depths at 5-meter intervals at 3 stations in the North Pacific, shown here in this heat map. 130 Synechococcus ASVs were identified, and those are amplicon sequence variants that represent distinct genetic content of samples using the CTD rosette pictured here. The overarching questions that are being asked here are, how does the ecology of Synechococcus change over oceanographic gradients? What environmental variables drive these patterns of diversity? And at what level does this diversity matter to the ecosystem? Bioinformatic techniques were used to analyze the data set using the R program. These are preliminary graphs that show evidence of depth partitioning. And here we have the SMILE data is across a strong temperature gradient at each station. And the abundance of Synechococcus with depth shows unique patterns at each station as we go deeper into the water column. And alpha diversity measures species richness or number of ecotypes present in an area. And as we can see here, the surface waters reveal strong gradient of diversity and kind of levels out as we go deeper in the water column here. And to further explore these differences in diversity, beta diversity was performed, and this measures the degree of community differentiation in relation to a complex gradient of environment. Here we can see that set 1, which is 26 degrees north, is more dissimilar from the other two based on how far they are from each set. And latitude and temperature are found to be significant environmental drivers of beta diversity patterns from statistical tests. Here are some really cool graphs that Dr. Algren provided, and it shows the community structure at each station here with depth, and also the beta diversity with the structure that is included. And this further shows why the southernmost station is dissimilar from its community composition that is less diverse than the other two stations. And upon analyzing these graphs, it was observed that the CRD1 shown in purple in these two stations are non-existent in the warmer tropical station here. And so we're hypothesizing that it prefers the cool, nutrient-rich environments. And this further investigation of these observations of partitioning were done through culture experiments in the lab. CRD1 and clade 6 are wild type representatives that were subjected to growth experiments in media containing different forms of nitrogen to examine the changes in growth that further explains the partitioning observed in the community structure. These cultures are grown over a course of 14 days and measurements were taken daily. Here are some zoomed in results of the second trial, and clade 6 seems to have more consistent sample readings within each media, but CRD1 measurements are a bit more inconsistent. So further trials and analysis are necessary to make concrete conclusions. And these results will further explain the utilization of nitrogen sources and how ecotypes of Synechococcus share spaces among related ecotypes. The overall project will provide insight into Synechococcus' partitioning characteristics and the mechanisms behind observed patterns. The microbes are not only important to the ecosystem, but also equally important to us humans, and are present in various fields of medical and health, environmental, engineering, the prediction of weather patterns, and also biofuel production. And thank you for listening. I'd like to acknowledge the support of Bill Exedo, McNair, the NSF, and NIH, and to Dr. Thompson for her mentorship. And then now I'd like to welcome any questions or comments. Thank you.
Great, thank you. Another excellent presentation. Any questions? So I'll go ahead and ask one again. So you mentioned, you know, they seem to prefer the cold, nutrient-rich environments, and I'm just wondering, do those things just happen to go together, or is it nutrient-rich because it's of the temperature?
Well, from like environmental factors, we know that the warmer waters are nutrient poor and the cooler waters are nutrient rich because of the oceanic currents that bring nutrients from the north of the cooler waters. And so we're kind of trying to see who is in what area, because I'm thinking that those particular ecotypes wouldn't do as well in the nutrient poor waters. They'd be like outcompeted is what I'm thinking, and other specialized ecotypes that are able to deal with like nutrient depleted waters would dominate in those areas.
Was there any like challenges in the research process that you faced or like analyzing the data? Just curious.
Yes, the our program was very difficult to navigate through Zoom, and every mishap seemed to take forever to try to fix, but we got through it. I'm definitely not an expert yet, but it is still in progress. I'm hoping to join some other people, hopefully in person this next fall quarter as we are getting back to campus. And also when we ordered our culture representatives, one of them was actually misidentified. So we had to start over and try to work with what we had.
Oh no, but I'm glad you realized that sounds
Yeah.
like a big challenge.
Yeah, before we got too far into it. So
Yeah. Great, thank you.
thank you.
All right, thank you for the presentation and congratulations on all that work. Okay, so we'll move on to our next presentation. This will be Gabriel, who's been working with Dr. Roberts. Ready to share your screen?
Yep. Good morning, everyone. Okay. So my name is Gabriel, and I'm currently a scholar in the Frias Maternal Fetal Medicine Lab at OHSU, and I've been working closely with Dr. Roberts in studying placental dysfunction. Before we get into our preliminary data, I'd like to give a brief overview of why the placenta is important. The placenta is a multifunctional organ that self-develops simultaneously as it provides the necessary nutrients and protection for fetal growth. I've included a schematic here that is a cross-section of the human placenta. And if you could see my cursor here, I'd like to note that there is a fetal side and a maternal side to the placenta, and both of these feature separate blood supplies that do not mix. The basic design of our study and preliminary data collection involves placenta that's been perturbed through the use of THC. We take that placenta from macaque models and employ immunohistochemistry techniques that can help us identify certain proteins of interest. In this case, we're looking for histone deacetylase enzymes, or commonly known as HDACs. HDACs play a pivotal role in post-translational modification of DNA and feature areas where and engage in areas that can covalently modify the histones which have the DNAs wrapped around them. The immunohistochemistry techniques that we employed involve the use of two antibodies that targeted our proteins of interest and amplified their signal in our tissue. And HDAC 1, 2, and 3 in particular have been identified as having roles in placental development, which is why we're looking for those proteins. And as for the use of THC, we've decided to use this model of perturbation because THC has become more and more prevalent in the use of pregnancy, but we still have very little understanding as to its effects long-term for mom and baby. And again, we're going to get into the results of our data here, but just a quick schematic overview, which you've already seen before, of the cross-section of the placenta. And here is a picture of one of the slides that I took. In the tissue itself is not as, it's more subtle, but the brown staining here indicates that we have found our protein of interest. Oh, no, it's not moving. Sorry. So here I've included two different magnifications of the tissue that we worked with. And the 60 modification gives us more of an overview of how much of the tissue was stained with the techniques that we employed, as opposed to the 40 magnification which gives us a more detailed view of the cells that we are looking for, or the proteins that we're looking for, where in the tissue they were found to be. And also to note that when we locate our tissue or our proteins, we can begin to investigate, well, what is the function that they play? And under this perturbation with the use of THC, has the role changed or how has it been modified? Here's a view of the HDAC3 staining that we saw. And I'd like to note here that the differences between HDAC1 and HDAC3, you'll see with HDAC1, the staining is more pronounced or more localized, whereas the staining in HDAC3 seems to be more discrete and across various cells. So as far as next steps in our ongoing research, we hope to employ other scientific techniques that can help us elucidate the role of the enzymes and how they're affecting placental development and how that translates to fetal development as well. So lastly, I'd like to take some time to acknowledge the Bill Isotope Program for affording me the opportunity to be involved in this research, and Dr. Roberts and JJ Torovius in particular, who have taken me under their wing and guided me along this process, and I'm so very grateful and indebted to them for doing that. So if you have any questions, I'll take them now.
Great, thank you for a very interesting presentation. Any questions?
I got one. I'm very interested in your research. Is up my field of interest, honestly. I just wonder why you guys didn't use a human placenta. I mean, didn't have any, is a lot of protocol to get one, or?
So the reason why we don't use human placenta in these types of study is because there's an ethical limitation to using human placenta and human tissue, if you will, in these types of studies, because we don't really know what the outcomes will be. So it's a lot safer to employ non-human primate models, which have similar architecture to human placenta. But because we don't really know what the overall outcome is going to be, we'd like to limit adverse effects in human pregnancies, which is why we don't use human placenta.
So I was just wondering, in looking at those slides, like what, I mean, we can see some visual differences, but how do you analyze that to demonstrate whether there's really a contrast between the normal and the THC version?
Right. So in the slides, you'll see more of a subtle difference in the staining, but some of the limitations of the immunohistochemistry techniques, basically they can tell us if the proteins are localized in the tissue and where in the tissue are localized. But as far as like understanding, what's exactly going on and why there was more staining over here or less staining over there, I think that's where we begin to think about what other techniques we can employ to understand what's going on in that specific tissue.
So this is sort of, it sounds like an innovative approach, and you're looking at something new, that trying new techniques. Understand this.
Yeah, so immunohistochemistry is, I think it's long been in use, but it's an ongoing study. So it's certainly, I feel as though, maybe my lab mates have a different view, but I feel like it's an exciting frontier medicine because, the placenta has not really been studied all that much, and there's a lot of work to be done, but certainly excited to take on that endeavor.
I have a quick question. I know at the beginning of your presentation, you mentioned that there was like a male and female anatomy or something within the placenta. Do you know what the difference between those are?
Yeah, so in the placenta, there's a maternal side and a fetal side, and they both supply different blood supplies. that shuttle nutrients back and forth, but they never mix.
Oh, okay, gotcha.
Great. Any other questions? All right. Thank you for another fascinating presentation.
Thank you.
So we're now to our final presentation, and we'll call on KJ, who's been working with Dr. Oriana.
So hi, everybody. I've been working with Dr. Oriana and some other Exito undergraduate interns working on the Chime In program, which is the local Portland chapter of the NHBS, the National HIV Behavioral Surveillance Program through the CDC. And the NHBS has annual research cycles. Oh, let's see if that'll advance. There we go. We have annual research cycles that focus on high-risk metropolitan areas and understanding the HIV transmission dynamics that occur within them. There are, I think, 25 sites operating currently, and pardon me. And every year they focus on one of 3 risk groups. They are the heterosexuals at high risk for transmission, PWID, persons who inject drugs, and MSM cycles for men who have sex with men. Typically, in a regular NHBS year, what happens at the Chime In program is that there's a 2-part data collection process. A big feature of the NHBS is that the program is conducted anonymously. So in the Portland area, it's the largest study in this region that allows us to collect information about people's HIV risk-related behaviors. This has to do with their partner network sizes, their personal relationships, substance use behaviors, et cetera, that can kind of shape their particular vulnerability to HIV. And we also collect information about how regularly they interact with HIV care and HIV testing. And it sort of runs the whole gamut of what people's risk and exposure profiles can look like. And then optionally, in parallel to that data, we conduct anonymous HIV testing, which then gives us an idea of what, who's getting access to care, who's getting exposed and getting transmissions, and monitoring that. And each of those 3 groups generally has a 1-year cycle. Our 2-part data collection process involves a formative assessment period, which is qualitative interviewing with both community participants and professionals that serve the cycle population, can be healthcare, social services, et cetera. And then we move to a quantitative interviewing period, which uses a core questionnaire developed by the CDC, and also local questions that are developed with community partners and other research institutions in the area. Because of the COVID-19 pandemic, at the NHBS, we have been in a 2-year MSM cycle during the time that I've been on here. And that has kind of allowed us to have a little bit of a deeper dive into some populations. And it's also sort of changed the structure. So we have preliminary data for both of the populations or subsets that we've been working with for this cycle. In 2020, the NHBS became all remote. So we did not conduct any HIV testing, and because of the structure, it allowed us to focus on a research question that had been several years in the works, which was expanding the MSM cycle to include transgender MSM in the survey, who currently don't qualify, but transgender individuals in general are an understudied risk population, and there isn't currently a good way to include and understand what kind of unique risks, care access barriers, and relationships to HIV care that this population might have in the Portland metro and other metropolitan regions. So a big part of our research this last year has been conducting qualitative interviews with community partners and transgender MSM in the Portland area. And then we've also been working on reintroducing HIV testing in 2021 for our, and that data collection is just now beginning. that'll give us a sense of what transmission looks like in in the pandemic context. So our key goals and questions have been generally for the TMSM population, what are the barriers and facilitators that can influence transgender MSM's participation in HIV surveillance research, as we are getting ready along with sites in Seattle and some other areas to introduce, oh, pardon me. Sorry about that. That was my own timer that was supposed to be silent. All right. Anyway, so yeah, there we go. So looking for what are the unique experiences that might encourage or discourage transgender participation, and also looking at the general transmission risks among MSM in 2021. And both surveys will collect information about the COVID-19 pandemic and its impact on, there's been a lot of care disruptions, but also a lot of changes to people's behavior. So we're looking at the net scope of that. And we do have preliminary data on the TMSM cycle, which has just been the continued development and outreach of relationships and information on how to ask questions and how asking questions of transgender MSM and doing outreach and just encouraging participation and what types of venues that they are most likely to connect with us through, maybe different than cisgender MSM, along with developing partnerships and clear and inclusive appropriate criteria for the inclusion of transmasculine populations. So that's all of that's the data that we've had a chance to collect during this year, and we will be probably within the next week starting actual HIV testing to get a sense of how that's worked in 2021.
Thank you for an interesting presentation. Any questions?
I have one. So you mentioned that you were on the MSN cycle for like 2 years. What's the next one that's coming up? I'm just kind of curious. It's an annual one.
Thank you. Sorry about that.
No,
But my audio actually cut out there for just a moment, but it sounded like you were asking about what the next cycle is going to be. The next cycle is going to be the PWID cycle. We haven't gotten, and I imagine that regardless of what the pandemic looks like, we will be moving into the PWID cycle next year, but exactly how that's going to look is going to depend a lot on how remote things are, and also probably the impacts of 2021 on data collection for the sites in general.
great. And you also mentioned that the COVID, like the changes because of COVID allowed you to, dive deeper into the transgender populations. Is that something that you think will continue because of this research to be more included in the next MSM cycle?
That's a good question, and I'm not 100% clear on whether that will be coming around for the next MSM cycle and actually show up as like CDC approved criteria for the inclusion of transmasculine people. But that said, that very much is the goal, is to get to that place. And one of the we haven't finished any like public facing research products out of the TMSM cycles data yet, but we have sent reports up internally to the CDC. And we're working at, like I said, Seattle is one of the other sites that's actively working on trying to figure out the best way to do that inclusion so that we can start collecting TMSM data as soon as possible.
Awesome. Thank you so much for your research.
Yeah, thank you.
Other questions? So I'm wondering, in this pandemic cycle, if the recruitment and sampling has been a challenge, and if you're getting similar sample sizes to before, and whether you're generating enough quantitative data for the transgender analyses.
That has definitely been one of the challenges. Getting materials during the pandemic has been extremely complicated. Now that we're working on resuming the HIV testing, the supply chains have definitely operated differently this year than in previous years for the medical supplies. And with the TMSM data, we did get a smaller sample size than was sort of hoped for, and smaller than compares to previous years of like MSM data collection for the cisgender population in the Portland metro. But so it's almost assured that both the pandemic and the smaller population size played a role. It's not totally clear, and also it being the first year of the NHBS operating in a remote context. In previous years, specifically the MSM cycles operated off of venue-based sampling. So it was very much an in-person cycle. And so moving all of that, having that be the cycle where everything became remote, sort of presented unique challenges just to having sort of continuity with partners and community spaces that usually helped it run and reach the number of participants that we were looking for.
It's clear it was a particularly challenging year for all of these reasons. Everything got disrupted. But as you said, it provided some new opportunities for looking at things that hadn't been examined before.
Yeah, definitely. It's been an interesting year in a lot of respects.
And Roberto leaving Portland too, huh? Another wrinkle.
Yes.
Right. Well, thank you again for a great presentation. Any other questions? All right. Well, that was really interesting for me to hear all of the presentations today. Again, I'm always so impressed by the type of work that you're doing and the level of sophistication and really tackling some important topics and generating really, really interesting research. So congratulations to all of you. It's wonderful to see what you're accomplishing. And it was also interesting to see people at very different stages. Some working on publications and some just at the initial stages of developing projects and getting things started. So a wide range of work. And I'll invite any of you to share your thoughts or comments about hearing the presentations from others in your cohort.
Yeah, I would say I'm a little bit further on in my research at this point. I was in cohort 4 previously, so I have a little bit more time. But for everyone else that's still going, like, don't get discouraged. Like, research, it's very difficult and it's like really disheartening at times. But just don't give up. And like, no matter where you're at in your research, like, just try to imagine where you're going to be a year from now. And like, I know you're starting my research is just like where I am now. Like, I never thought that I was going to get to this point. Like, so many failures in my lab happened and setbacks and just like made me want to cry in the basement of the chemistry department. Just like, so it was a lot. So just like, don't let that bog you down and just keep going. And remember that like you are doing something that's not only going to benefit like yourself and your PSU community, but the greater good of like science and humanity. So just try to keep that in the back of your mind the entire time, no matter how tough things get.
Right. Perseverance is important, especially, in a pandemic. So again, congratulations to all of you for working under these very unusual circumstances. Okay, well, it's great that most of you, if not all of you, are going to be continuing with your RLCs as we enter the new school year. So you'll be able to continue this great work, and I hope that we get to see future iterations, as it reaches completion, and you, maybe are doing future presentations and publications. We're excited to see it get to that stage. again, I want to just thank all of you for being part of this program and showing what great results we can get when undergraduates have time to get involved in research and really make contributions to your labs and to your projects. So thank you for the work you've been doing, and want to wish you the best in the new school year. So with that, I guess we can wrap things up, and you can all now breathe a sigh of relief. You've been through the symposium, so another milestone for Exeter.